I'm Phil, I am a microbiologist, and I appreciate the title of my talk is slightly out with the main theme of this conference, but it's when we launched the Vaccine Development and Evaluation Centre last year that Paul phoned me up and said he thought the subject matter would be of interest to you, so hopefully it is. And just to reassure you, we do also work on antimicrobials, diagnostics, therapeutics, and other medical countermeasures. We just couldn't cram that all into one nice little acronym like VDEC. Hence, that's the title of my talk. So hopefully you'll be aware that the UKHSA was formed during and out of the pandemic, and part of its remit is really to learn the lessons from the remit, particularly in response to the fact that what we showed was when academia, industry, government, and other non-government organisations work together, we could achieve much more than the sum of our parts and actually get those medical interventions to patients, which eventually is what it was all about. So that's the overall remit of UKHSA. We were lucky enough during the pandemic to get some funding from what was then called the Vaccine Task Force to actually design and build some brand new state-of-the-art laboratories at Porton in Wiltshire, and also refurbish some of our older laboratories which date back a little bit further. So I just want to touch on Porton, because when you mention the word Porton, lots of people have concepts about what Porton Down is. So it's actually a geographical location 10 miles north of Salisbury. The main centre of it there is this bit up here, which is the Defence Science Technology Laboratory, which is part of the MOD. That's been there for 100 years, and its main remit is to protect our armed forces from CBRN and other threats. They do a lot of cyber and other stuff. We have been there since 1940 as a microbiological research establishment and been very much part of the Department of Health Family since the 1970s, studying and understanding infectious diseases and how that affects the UK population. There's also a science park there, the Porton Ranges, where they can test all sorts of things, including the dragonfire that you might have heard of on the news this morning. But in general, it's always referred to Porton Down. So when we launched VDEC, we invited the press pack down. We invited them behind the wire. They were very excited about that. And that did generate a number of headlines. So the first one here, UK scientists begin work on developing against disease X. To be clear, we are not developing disease X. We're developing the plans to fight whatever the next pandemic might be. This one, we want to stop the next pandemic in its tracks in 100 days. Again, just to be clear, we are not developing the vaccines. That's the job of industry to actually manufacture the vaccines. But what we can do is help them test it. And then Emily, our young scientist there, got very excited about this next one, Secret Labs vaccine plan. She said to me, Phil, why are they calling us a secret lab? We invited them all in, we showed them round. It's the press, Emily. But we are concerned about potential pandemic flu, avian influenza, etc. So what is it that we actually do? We work across this pathway from discovery to delivery. And we work in certain parts of it, not all of it. But as I said, we work with academics, industry, NGOs, governments, etc. Initially, when there's a new pathogen identified or if one starts spreading, to do the isolation, the characterization, etc. We can then move forward into developing some assays in preclinical models, so that's animal models, understanding the routes of infection, developing challenge studies, assessing immunity before things go then into the patient in the clinical trial. Again, we don't do the clinical trials. That's the clinical research organisations and the clinics that actually run those clinical trials. But we can help develop qualified assays to the appropriate regulatory standards, so GCP, good clinical practice, to help support those vaccines and generate data on the immune responses, for example, to the vaccine. And then also we do some post-licensure. So one example is that the AstraZeneca Fluenz Tetra jab, the intranasal flu spray every year, we test every batch of that is produced in the UK and release that for them. So it's very much working in partnership across this pathway. So digging down into some of those details, The preclinical, we've been working in preclinical in animal models for many years, and actually we've been working internationally for many years. A lot of our funding actually comes from the US government because the US government's got a lot of money, deep pockets. So we are an approved partner for BARDA and National Institute for Health and NIAID, US government-funded organizations to develop those animal models. During the pandemic, we also worked with CEPI and to support their 100-day mission of having that new medical countermeasure ready to go. Again, we're an implementing partner for them for providing support into the development, but we're also part of their central network of labs. So during the pandemic, we developed a microneutralization assay using the live virus so that we could assess patient sera from the clinical trials of the vaccines they were given or people who'd been exposed. And if any of you were actually part of this SIREN study, donating blood samples throughout the pandemics, many thanks for that. We were able to take that into our diagnostics labs. And then carrying out a range of tests against medical countermeasures on different vaccines, whether they were the vector-based vaccines, the mRNA, protein-derived vaccines, as well as antivirals, therapeutics, monoclonals, and even passive transfer. So having established animal models, you also want to think about the patient and where we come into things. So moving away from this into the sort of diagnostics sphere, we were tasked during the pandemic also to help industry with its development of diagnostics. And this is where the SIREN study comes in. We've got over 100,000 blood samples stored at Porton, and we can do lots of analysis, and did lots of analysis on that, and look at the infection patterns over the course. But we also worked with the manufacturers who were developing the lateral flow devices. I think it was 196 different devices were sent to us for analysis, and we put them through a triage system to work out their specificity, sensitivity, and reproducibility, and we did that on live sera. Once we'd gone through that triage, there were actually only 5 that passed the test, and then we could advise Department of Health to actually then issue procurement notices. They were purchased, and then we all got used to using them on a regular basis to control our workforces. We are still working with those manufacturers as new variants of concern come up to test them. The good news is that, yes, those lateral flow devices are still detecting the variants of concern. A couple more have now been licensed for use. And we're also talking to the manufacturers about what next. So can we put flu antigen tests in with the COVID ones? Can we put RSV in? We did talk to them about Mpox. They weren't quite so interested in that. But in that case, we can then develop some of the assays in our labs so that when things like Mpox do come along, again, we can skill up and do some rapid diagnostic of those emerging diseases. And then also work with the manufacturers to try and put them on some of the large platforms like the Roche Cobas system that you see over there, and some of you might be used to having in your hospital labs. And then the sequencing, taking patient samples and running the sequencing. And there we're very much part of a network across the country that did almost 3 million sequences and is continuing to do so. And some of the latest research is actually taking then patient samples off those lateral flows and putting that through the sequencing platforms so that we can keep a track on novel and infectious diseases as they emerge. But it's not just all about pandemics. We realise that also there are the common or garden microorganisms, if I can call them that. So again, we have research teams working on some of the organisms that are in the routine immunisation schedules, or some of the ones which are posing clear and present dangers. Developing correlates of protection, helping companies design vaccines or reconfigure vaccines to these more normal organisms. Looking at things like biofilms that we heard about yesterday in UTIs, mucosal vaccines, and also maternal vaccines, and passing that protection on to the infant. So research going on there. In VDEC itself and in the facilities that we built during the pandemic, this is an overview of what we did. What you can see top left is a class 3 microbiological safety cabinet in a containment level 3 laboratory where the scientists there were actually growing live SARS-CoV-2. I took that photo, so I know that's the case. When the press came down, we didn't let them into those labs. We only let them into clean labs, just to reassure you. And we've now got us 2 purpose-built buildings, as well as our original main building, in which we've got suites of BSL-3 laboratories to do the live virus isolation. We can then inactivate it and bring it out into a BSL-2 lab and analyze it on platforms like the CTL scanner that you see bottom right, with all the associated freezers, equipment, everything is monitored, etc., to those GCP standards, so that the data that we generate from these studies can go back to JCVI, to the MHRA, etc., for the licensure of vaccines. And the main thing is that we have the ability to do multiple platforms. So this is a similar summary slide. Early days, isolate, identify, qualify the pathogen. We can also generate quality control material, if you like, provide formalin inactivated material or X-ray irradiated material so that can be sent to other labs who can study it at lower containment levels. Very much working with the developers. So for CEPI, I've mentioned the central lab network. What we've done is that microneutralization assay that we developed during the pandemic, we have now tech transferred that to accredited CEPI labs around the world. So those include ones in Europe, Southeast Asia, and Africa, so that the partners there can analyze the spread of the virus in their populations, but also the impact of vaccines in their populations, because that's very much part of the CEPI mission, getting a quality of healthcare out to low and middle income countries, and we very much support that. We did lots of trials in the UK with the ComCov COVID different trials for NICEC, part of the Department of Health, but then also worked with some of those industry partners to analyze those on a fee-for-service basis. As I said, the main thing is to have the ability to have qualified, validated assays against a range of organisms, whether they be the routine ones in some of the pediatric vaccination schedules or some of the new and emerging targets. And I think I should probably add onto this list Lassa. We've just been tasked with coming up with a qualified assay to Lassa, should that be a potential future issue. As I said, it's not just vaccines. We also do a lot in the antimicrobial space, and this is a group run by Mark Sutton and his team, where, again, they're helping industry develop novel antibiotics, look at novel mechanisms of action, whether those include efflux pumps, novel peptides, phages. Again, yesterday we heard a little bit about phage therapies making a bit of a comeback in certain circles. And we've recently joined with UKRI to be part of their PACE program, which is very much driving that academic look for novel mechanisms because of the antimicrobial resistance issues that we've been hearing about, and to help develop antimicrobial stewardship. And again, a range of scientific practices to enable us to understand the organisms, work with the organisms, look at how novel antimicrobials might actually affect them in situ. This slide has a couple of other examples of that. The one that I would probably draw your attention to at the bottom is actually an impedance-based method that we've developed with the University of Southampton. This is for near patient sample testing, particularly in urinary tract infections, where you can take a sample, put it through the impedance, and very quickly get a readout of what organism that patient has and what its antimicrobial susceptibility profile might look like. So if any of you are interested in developing novel microbials, do come and talk to us, and we'd be happy to welcome you down, and even can host people working in our labs to further this type of research. Moving on, the penultimate thing I'd like to speak about is the built environment. Again, this is a new facility that we have designed and built within the last couple of years. Our biosafety team have a lot of experience in working in aerobiology. So this is Ginny Moore and her team, but we have now built this mock hospital ward. So as you come in, you've got a sort of standard nurse center. We've then got some sluice rooms, a couple of rooms with washing machines in them. And here we're looking at things like, what is the difference between washing scrubs in a traditional, that's my timer, a traditional commercial washing machine, or using a domestic washing machine? And with things like the energy crisis, are people taking their scrubs home and actually turning the washing machine down to 30 degrees, etc.? What's the infection control risk of running those things? Those are the sort of experiments we can do there. We've got a couple of isolation rooms, single isolation rooms with en-suites, built to exactly the same standards as you would find in the NHS, and then a 4-bed ward at the end. And again, we can do infection transmission studies in this environment in a controlled scientific manner, as opposed to going into hospitals and disturbing patients. So again, one example, during MPOX, we did have teams going into wards and looking at what was the effect of once a patient comes out of a particular isolation ward, the health staff then go in and change the beds. Does that aerolise the MPOX? Is that posing a threat to the healthcare worker or for the patient coming back in? How many air changes do you need, etc.? So rather than doing that in Luton, we can now do that in our model ward. So again, and there's lots of companies here with different surfaces, different disinfection techniques, etc. Those are the sort of studies that we can do. And then finally, the last thing I wanted to talk about was training. So we have a novel and dangerous pathogens training team. We've done laboratory training for many years at Porton. We now have a brand new facility there. This is our latest cohort of sandwich students that we were putting through some microbiology training. I don't know if any of you out there were involved in the UK's response to the Ebola outbreak in Sierra Leone from 2014 to 16. But if you were, some of you might have come down to Porton for the training. We trained a lot of our staff that were deployed. We trained a lot of NHS staff who were deployed, stood up and volunteered. And that's the sort of work that we can do in there. And things like safe training for changing of gloves that, Lindsay, you were talking about in operating theatres, but doing that in a situation where you may be exposed to blood-borne viruses, etc., but in a safe environment. We use fluorescent markers, for example, so there is no infection risk in these training labs. So NADP, we offer that as a course. So hopefully that's a very quick run through of what the UKHSA capabilities now are. We're only part of UKHSA, but to recap, we have the high containment capabilities, we have the modelling, we can work to the appropriate quality standards, and it's very much that partnership model that we're interested in to prepare, develop, and build for whatever Mother Nature throws at us next. So top left, you can see the cartoon of the buildings, but at the bottom there, you can see sunrise over the new labs as they exist. So thank you very much.